Now, besides the really important ideas of genetic modification that we talked about with the bacteria and insulin, there are plenty of other examples, but I'm not going to spend as much time talking about them, and I don't want to make you listen to too much more. But I will encourage you, if you're interested, to read the rest of these slides, and I'll just kind of go through them quickly. So, first of all, you might have heard of genetically modified organisms, or GMOs. These are organisms that have been genetically modified, again, using the idea of pasting a desired gene from one species into a bacterial chromosome, and then maybe putting that bacterial chromosome into the tissues of a plant or an animal and getting that plant or animal to have a trait it didn't have before. So in the United States, we commonly do this with our food. It's less common in other democratic nations, such as in Europe, and that's just because of the way they vote. But essentially, we've used GMOs to get plants to manufacture their own toxins that really were evolved in bacteria. We can get these plants to produce bacterial toxins so that we can kill bugs without having to spray pesticides, and that protects the environment from all sorts of toxins. We've had artists work with scientists to show that we can transfer glow-in-the-dark genes from a jellyfish to a rabbit to get to glow in the dark. That is nonfiction, not fiction. We've had companies want to take genes from spiders that make spider silk and put them into goats so that goats can make tons and tons of spider silk, so that spider silk could be collected to make really lightweight but super strong body armor for soldiers, such as those in Afghanistan. Now, since 1990 and up until 2003, we had all sorts of different governments worldwide working together to try to complete the so-called Human Genome Project. And the Human Genome Project was an attempt essentially to figure out what are all the genes in the human cell and what proteins does each make. We still don't fully understand all the results of the Human Genome Project, but the idea was if we can figure out what all the genes are, where they are, and what they do, then we might be able to genetically modify humans, or at least come up with medicines that would treat their medical diseases. Now, this diagram is far too complex to explain right now, but I'm sure a lot of you have heard of CRISPR-Cas9, which is a system that's been recently engineered to allow us to edit the genes of just about any organism you can think of, including humans. And this is a slicker way to do it than with those bacterial plasmids we talked about before. Again, I'm not going to explain this whole thing to you, but we learned about it from what bacteria naturally do, and then we've engineered an equivalent that can allow us to remove genetic diseases from humans and, maybe even get those humans to have different traits that shouldn't matter that much, like eye color, hair color, height. We could edit those in our future babies. More on that later. And of course, we have to talk about laboratory cloning. Laboratory cloning is, of course, different from natural cloning. If you know any identical twins, they're natural clones of one another. And you don't want to say you're against cloning if you're against laboratory cloning. Otherwise, you've hurt those people. So laboratory cloning is essentially when we take an organism that's already alive, choose to try to make a genetic copy of it. And this has been done since 1952, but we really first got a lot of attention when this was done with mammals. So most famously in 1996, when they did this with a large mammal, such as a sheep, but it was done earlier to the mouse in 1986. So the basic technique is essentially the following. You take an organism you want to clone. Let's say this man and woman are married, but let's say she has a devastating genetic disease and she doesn't want her child to have it. Maybe the two of them decide they want to clone him. They're really into him. So the way they can do that, it's intellectually simple, technically difficult to pull off. But like I said, this thing has been done with the rest of them. So here it goes. We could surgically harvest her unfertilized eggs from her ovary and basically get rid of her DNA. Because as we said, they don't want her DNA involved since she's really sick. So they get rid of her DNA. But what they do is they harvest one of his cells, and they take the DNA from his cell and put it into her unfertilized egg. That tricks that egg into believing it's fertilized. So that fertilized egg now, or sorry, that pseudo fertilized egg has all of his DNA and none of hers. In the petri dish, we can get that cell to reproduce itself. So you get a small ball of cells that are all genetically identical, and we could implant that into her womb at the right time of month to trigger what would after that be a normal 9-month pregnancy. So when she gives birth to the baby is her husband's genetic twin. Now, she might see that baby's parents as her and her husband, but really genetically that baby's parents would be his mom and dad. So those are all biological facts. They might seem a little socially awkward, which is why laboratory-based cloning of humans has been banned by most advanced governments. But it's theoretically possible, and I wouldn't be shocked if it happened sometime. Now, if you're really creative, you might wonder, hey, is it possible to make a new species that hasn't existed before? Yeah, you could. So if you're really fascinated by that, you might want to read over this slide on your own, perhaps type in this link and check out a little video about it. But yeah, humanity has created a species that didn't exist before through genetic manipulation. So this is part of your life, and as you become an adult, you're going to become much more aware of it. Finally, just, I don't expect you to really take all this in, but this timeline is already well out of date. It's just the coolest timeline I could find. But what we've known about DNA goes back to a scientist that you might have learned about in middle school, Gregor Mendel. He figured out the difference between dominant and recessive genes by doing genetic problems based on pea plants. And the body of work has just gradually built, and it's becoming more and more fascinating, and it's accelerating faster and faster. And I'm really excited to take you to the present, past 2003, and to the present and beyond. So DNA is so important because it's the code that runs the cell and tells it how to make the proteins that matter to it, whether those proteins be hormones, enzymes, or structural proteins. Take care now.